What's up, guys? We are back. Part number 3 of the sequence of biology questions that we're going to be exploring for the IMAT. So, once again, I'm going to ask you to try to sort the questions on your own first, and then we can explore it together. If you ace it all of them, that's great, and it's okay if you have difficulty with one or another question. I hope the explanation will help you, and if it still doesn't hit you properly, just shoot a question in the comments or just reach out to me on Instagram, for example. Okay, so let's go. Question number 1 or 21. Remember that this is a sequence of videos. I have my little playlist here, and we're building into 10, so we can cover 100 questions. This will help you to prepare to what's coming next into the IMAT biology questions. Okay, first question. Put the following structures in order in which they function when Jonas sees a bowl of chicken and moves towards it. So it's a little context to talk about the nerve system. Basically, this question I want to know if you understand the order of the events and how this influences us. Okay, so basically, if Jonas is seeing a bowl of chicken, there's a visual stimulation that first goes through the eye. Okay, we have this little structure here they call retina, or whatever it's pronounced in English, and this is directly connected to our brain to neuron that we're gonna call it a sensory neuron. Okay, so it doesn't do anything but just drags information, a message to our brain. Okay, so far we have number 1 and number 3. After that, the brain get the message, say, okay, I know what you want, let's go grab it. So there will be an action going on. Okay, so our muscles are going to grab that bowl, or just gonna walk towards it. But before the muscles can take place and to action, we have to have this information go to them. Okay, and that's gonna happen by the motor neuron. So let's just recap. First, we're gonna have the eye contact, the retina, 1, the sensory neuron, 3, brain, 4, 2, and 5. Do we have this option here? Absolutely. It is letter E. Okay. But basically straightforward. Not much to say about this one here. It's just remembering the structure, the basic structure of our nerve system and how it works in order to accomplish a mission. Let's go to our second question. Oh, this one is interesting. It's talking about clone. This usually is a topic that we don't usually study in our preparation to the IMAT, because we don't usually think that this can be asked, but it is. It is probably going to be asked this year. Usually, it is in all years, and the official documents that talks about the topics from the IMAT. So biotechnology, it's there, and basically cloning, nothing more is than biotechnology. So let's check these following statements and try to identify the ones that are correct from the ones that are incorrect. The first one: animals cloned from embryo transplants or genetically identical. Yeah, that's basically what it is. Even when you think about the word clone, it's basically when you have one thing and then you duplicate it into something that is exactly the same. We have a lot of films, we have a lot of series and fiction stuff talking about it, because it's very interesting, very exciting to think about this. So, statement number 1 is correct. Second one: the genetic material is removed from an unfertilized egg during a adult cell cloning. Okay, so this is a little bit more deep. It goes right in the core of the understanding of clone, and this one, the people that didn't really got to study this topic will be a little bit confused. But statement number 2, it is correct, because basically how it works is that we're gonna have to work with 2 different cells. Okay, first you get the individual, the animal, the person that you want to clone, and then you just select a cell. Okay, so it can be from anywhere of our body. We're gonna have an adult cell. Okay, so it's important to be an adult cell because we want the whole information there. We want the information that came from the mom and the dad, and that makes the person or the individual just the way it is. Okay, so it's gonna be an adult cell. And then secondly, we're gonna need an egg from the partner, let's say, but it doesn't matter. the important thing is that this egg is not fertilized, okay? So we're going to remove the material, the genetic material that it is in this egg, and we're going to use this egg as a shelf, okay? We're going to have all the structures inside of it, all the things that we can find in the cytoplasmatic of a egg cell, and we're going to introduce just the nucleus of the adult cell. So we're going to have this Frankenstein. We're going to have this cell that is young and all the cytoplasmatic information, but also old in the core, in the nucleus, okay? So what is going to happen is that this young egg is going to have the potential to multiply because it's going to think that it's fertilized because it has the whole information in the core, in the nucleus. And while doing that, basically all the characteristic of this individual will be just like the adult that we cloned because it has the same information, okay? Same genes leads to the same person. That's how twins works most of the times, okay? So this is very crazy to think because it makes the cell itself a little bit confused, and this is one of the reasons why we don't talk much more about cloning, because the first time they did that, it didn't really work well. I don't know if you guys heard about it or remember or just had a chance to study, but the first individual, the first mammal to be cloned was a sheep called Dolly, and unfortunately Dolly passed away very quickly. She had the chance to develop into an adult, but a couple of months later, it died. It had a premature death. And that happened because the cell was confused. The cell was behaving as a young individual, organism, the cells of that body, but at the same time, the material DNA that it was inside, it was an old one. And this is a small detail that usually we don't talk about it when we're explaining it to mitosis, is that when you get the chromosomes of the cell, we're gonna have a little part, a little structure, a little section of this chromosome in the edges, okay? Just remember that chromosome ready to be split, okay? We're gonna have this little structure, and the very edges, we're gonna have a structure that we call telomeres, okay? And the telomeres is very important because it's going to be the clock of the cell. By this structure, the cell will be able to coordinate itself to understand that, okay, it's time to split. It's time to not split. And once, just trying to explain very briefly, but once you have this clonation happening, it kind of loses the orientation, and then the individual start multiplying itself many times, and this leads to a lot of disease and tumor and cancers, and usually the individuals that are cloned don't survive long, okay? So I'm gonna leave a link in below to explain this a little bit better than I do, but important thing here is to understand that statement 1 and 2 is correct so far. The third, cloning cause reduced gene pool. Perfect. Basically, gene pool is just like a metaphor or whatever to say that we're gonna have this space of possibilities, space where we can have different characteristic due to the variability of our genes. Okay, when you have a clonation, we're gonna have two individuals exactly the same, so there's no much space for differences. The only difference that can happen, for example, is if it's this individual that is the same as this one, this had more mutations than this one because of whatever, one smoked, one doesn't. Imagine a sheep smoking, oh my god. But the third one is perfect, it's correct. So they're exactly the same, so the variability it's shrinked. Okay, so fourth statement, cloning is only possible with mammals. Nope. Basically, we do clone a lot, especially when it comes up to biotechnology, but mostly with bacteria, okay? But it's also possible to happen with other classes of animals, okay? Not just mammals. So this leads us to 1, 2, 3, letter E. Okay, next one. Oh, this one is important as well. It's about physiology. So it wants us to identify and organize the path that the blood takes from the kidney to the liver. So it's important to remember that the kidney is one of the last pit stop that our blood goes through before leading to the heart. Okay, so if it's going from the kidney to the liver, it has to be already leaving the kidney, and that happens through a vase. Okay, and every vase that goes towards the heart, it's going to be called a vein. So if let's check all the veins option here. Pulmonary vein, no. Inferior vena cava, no. Aorta, no. Renal vein, perfect. So we're going to start with this 6. So the renal vein is not going to go straight to the heart because we have 2 important collectors, okay, big vases that it's going to be called vena cava, okay. So if it's coming from the inferior part of our body, it's going to be called inferior. If it's coming from the upper part of our body, it's going to be called superior. So 6, 2, after that it goes to our heart, to the right side, to the trying to remember the translation in English, but to the arterios, right side. But this question doesn't really ask us about this, only when it comes up to the vases. So remember, from the right side of the artery goes to the ventricular right side, and then from there it has to go to the lungs because it's a deoxygenate blood. It's a blood that coming from our body. Okay, so every phase that goes away from our heart is basically called artery, as you guys know. So let's identify the options that we have. Artery, pulmonary artery, perfect. So 6, 2, 1. After it's oxygenized, it has to go back to the heart. So let's search for a vein, pulmonary vein. After it's in our heart, it has to leave our heart. So aorta, okay, this is the big port of X from our heart. And after that, it will eventually find its way to the liver. So hepatic artery. So basically 6, 2, 6, 2, has to go 1, 5, 4, 3. So letter D, that's what we're looking for. Was a little bit rushed, but the most important thing here you have to take is that if you did understand this question, it's probably because it might be a little bit confused the path that our blood takes into our body. But the core understanding here is to understand how it flows into and out to our heart. Okay, so it's important to recap this section here. Let's move forward. If you have any questions, just don't bother to ask. Let's help each other. I can share some study material as well. So let's go. Key evolution concepts. So let's talk about evolution. It's always good to talk about evolution. Which following the statements are true when regarding to evolution? First statement: individuals within a species shows variation because of differences in their genes. Yeah, basically that is how it happens. So we're gonna have the differences, the variations, due to different genes. Remember that the genes are the sections of our DNA. There's a responsibility to express a characteristic. Okay, so the color of our eyes, the colors of our hair or skin or everything, our height. Sometimes it's not just one gene that's going to determine that, but a couple of them. But the genes plays a role in the variation. Second one, beneficial mutations will accumulate within a population. Perfect. So the evolution, basically, it's just a result of several mutations that we can find in our bodies. Some of them are good, so they maintain because they're going to be advantage for that individual, so that individual will survive a little bit longer and will happen to reproduce and spread that characteristic. And some of these mutations are bad, and unfortunately this individual will probably die sooner than it should. So only the good mutations will survive. Third statement: genes differences are caused by sexual reproduction and mutation. Perfect. So, and when you're thinking about individuals, the differences that can happen to him is due to sexual reproduction. Okay, so if he's not a clone, he will have differences in his DNA compared to his parents, let's call it this way. And more importantly, when it comes up to evolution itself, the mutations. Okay. Fourth statement: species with similar characters never have similar genes. Remember, all the sentences that we have words like never, always, only, these statements are very likely to be wrong. Okay, but by reading this, we can deduce that is just not correct, because species with similar characteristics, they have similar genes. It's like twins, for example. They have very similar. The clone, for example, they have the same genes, so they have similar characteristics. So, nope. This leads us to 1, 2, 3. Letter E once again. This is a coincidence, I swear. Let's go to our fifth question. Correct genetic statements. Once again, let's try to identify which statements are correct. First one. Alleles are similar versions of different cells. Alleles are similar versions of different cells. This statement doesn't even make sense. It's important to remember that we have these genes that's going to determine our characteristics, and these genes can have different ways of expressing itself, can have different variations, and these variations we're going to call alleles. Okay, so these alleles usually they are shown by pair, so we can have two different alleles, but some characteristics can be result by 3 or 4 or 5 different alleles. Okay, like for example the bloodstream, it's determined by 4 different alleles. Okay, so statement 1, nope. Second one, if you are homozygous for a trait, you have 3 alleles the same for that particular gene. So not necessarily. Okay, if you're homozygous, that means that almost the same. Okay, you have same something for that trait. Okay, so same alleles for that trait. It can be 2, it could be 3, it could be 4. So I would put this one in standby and check the other ones and see what could we match and accommodate. Okay, so this is not entirely wrong, but at the same time, it not necessarily means that we have 3 alleles. Okay, so let's move on. Third, if you are heterozygote for a trait, okay, different, you have 2 different alleles for that particular gene. So okay, that seems like correct, because most of the cases we do have 2 different alleles. Okay, alleles for that trait. We, as I mentioned, we can have more, but most of the time it's 2. Fourth, to show the characteristic that is caused by recessive allele, both carried alleles for the gene have to be recessive. Okay, well, basically what it's saying is that to recessive trait to be expressed, both of the alleles has to be recessive. Okay, so that's why it's considered recessive. So these alleles has to be expressed in a recessive way. That's basically what happens. This is the basic genetic statements from Mendel. And so maybe 2, 3, and 4. What do we have here? We know that 3 and 4 are correct, but we have no 2 along with it, so just forget about it. Sometimes you're gonna be a little bit confused, and it's important to use these strategies to not get like lost and stuck. And if eventually it's not matching, let's go with the closest one we have. So letter E. This is what we're looking for. Let's move on. Another question of physiology. Okay, this one is very interesting actually. It talks about water deficiency. So let's put the statements in the correct order of event for when there's too little water in the blood. Okay, so basically what does our body do when it's in a stress situation? Because too little water, lack of water in our blood, it is a stress situation for our body. So okay, what do we have here? Urine is more concentrated. Pituitary gland released ADH. Blood water levels return to normal. Hypothalamus detect too little water in blood, and kidney affects water level. So let's put this in order. important to remember that hypothalamus is the responsible to produce the ADH. Okay, so it's the antidiuretic hormone. So it's the hormones that's going to stop us from peeing. Okay, so the hypothalamus will produce, but it will storage in the pituitary. Okay, so it all starts with the Ford. He's going to detect and produce, and thankfully he will probably already have the ADH storage in the pituitary. So he's going to command the pituitary gland to release the ADH. So 4-2. After that, the ADH will give an information to our kidney to affect the water level, to make our kidney stop and absorb more water. Okay, this will make our urine more concentrated, and after all, what we're looking for, our blood water level will return to normal. So in this order, that's what we're looking for. So 4-2, 4-2, 5, 4-2-5. Yeah, this is the only option. 4-2-5. So letter C. That's our answer. So a very interesting fact, I don't want to make this longer, but when we drink alcohol, for example, the alcohol has a chemical reaction with our body that makes our ADH stop working. So it kind of stopped and blocked the ADH effect, what makes us wanting to pee more. And that's why we usually pee a lot. And if you don't drink water while you're drinking alcohol, you're going to wake up very bad, hangover next day, but mostly because you're dehydrated. You need to drink a lot of water. So after a certain age, it's important to balance the beer with the water. A little beer, a little water. A little beer, a little water. Yeah. Let's move on. Question 27. So, which of the following are true of the cytoplasm? I even put a little picture of one of the books that would be very important in the first years of medicine. It was important for me in biology course, and it's definitely going to be important for medicine as well, from Alberts. So, what is true about the cytoplasm? The vast majority of the cytoplasm is made up of water. That's true. That's true. 80% of all the material in our cytoplasmatic is water. The other 20% is going to be proteins, organelles, and etc. So, second statement, all content of animal cells are contained in the cytoplasm. So, everything that an animal cells has is contained in the cytoplasm? No, we do have the nucleus, the nice nucleus here. We have the DNA, for example. DNA is not in the cytoplasm. It's organized in this organelle that we can call the nucleus as organelle, for example. So, second statement is incorrect. Third one, the cytoplasm contains electrolytes and proteins. Just like I mentioned, the other 20%, more or less, it's basically organelles, electrolytes, and proteins. So, 1 and 3, what letter gives this us? Letter E once again. Let's move forward. We're almost there. Question 28 was supposed to be 28 here. So, meiosis facts. So, which of the following statements are correct about meiosis? Once again. So, the DNA content of gamete is half that. Sorry. The DNA content of a gamete it's half that of a human red blood cell. So basically, what it's saying, a little bit confusing, but that blood cell, the DNA of a gamete of a half. Okay, so let's check the true statements. The first one says that the DNA content of a gamete is half. Okay, so let's check the statements of this one here. The DNA content of a gamete is half that of a human red blood cell. Okay, a little bit confused, but it's just comparing the DNA of a gamete and the DNA of a blood cell. This is a tricky one. It's important to remember that the blood cell does not have a nucleus. That's why it has that strange shape, because it doesn't have the nucleus. Okay, there is a cell that originates the blood cell. That one does have a nucleus, but the blood cell itself does not have. So it's completely incorrect. The blood cell has 0 DNA when compared to a gamete. Okay. Second one, meiosis require ATP. Remember that ATP, it is the energy that is going to be required for most of the activities in our body, and meiosis is a very important activity, so it does require ATP. Second one, it is correct. Third one, meiosis only take place in reproductive tissues. That's true. So we're only going to have the meiosis going on in our follicles, for example, or sperms. So these are important because they're going to reproduce. Okay, and in meiosis, a diploid cell divides in such a way so as to produce two haploid cells. If we read it if you read it very fast, you're gonna probably think that is correct. Okay, so yeah, meiosis is going to diploid to double the DNA, divides into two other haploids, so half of the DNA, but it's not correct. It's important to remember that the meiosis is so, like, the way the meiosis happens, it has to first happen mitosis and then the meiosis itself. Okay, so the meiosis has these 3 stages. So in the end, we're gonna have 4 haploid cells. Okay, so only statements that are correct here is going to be the second and the third. So letter D. That is what we're looking for. If you're a little bit confused about this, I highly recommend you to review the meiosis topics because it's going to be very often in the exam. So 29, a question about the pH. So the pH of venous blood is 7.35. Which of the following is the likely pH of arterial blood? So it's important to remember that the venous blood carries a lot of CO2 because it's a poor oxygen blood stream. Okay, so it wants us to try to guess what will be the pH of the arterial blood. So basically a vase that has a lot of oxygen. So it's likely to think that it's going to be a little bit higher. Why? Because the amount of CO2 in the venous blood, it's going to be a lot, and this can react with water and produce acid. So this acid will kind of lead our pH a little bit lower. Okay, so the only option here that is a little bit higher than 7.35, it's the letter E. So this is what we're sticking with. You will never find our blood with these pH. This is too much. It's important to remember that our blood is tamponate, which means that we have structures there that is going to control the pH of our body. Okay, so letter E, perfect. And last one, let's see. Which of the following statements are true about asexual reproduction? So basically, reproduction that we don't have exchange of DNA. Okay, so the first statement, there is no fusion of gametes. That's true. We don't have a fusion of gametes. So 2, there are 2 parents. Think about bacteria, for example. It's going to reproduce asexually, so probably it's just going to be a mitosis. Okay, so there's no parent. There's one and there's the second one. Okay, so there's like it's a horizontal relationship. It's almost like a sibling, a sister. Okay, so there's no parents going on here. Third statement, there is no mixing of chromosomes. That's correct. There's no mixing of chromosomes. We don't have exchange of DNA. We don't have variability, so it's going to be the same DNA. And fourth, there is genetic variation. So there is no genetic variation. So once again, we are not going to have this because asexual reproduction is it's basically a mitosis. So this leads us to 1 and 3. Do we have this option here? 1 and 3, letter 8. This is it. Okay. I was trying to make this video a little bit under 30 minutes. So if something is not clear, if something's not clear, and if you have a little bit of not understanding of some topics, just drop me a message. Let's help each other. I'll be soon bringing more questions about biology, and well, hopefully enough, we're going to be prepared to the IMAT exam. Okay, if you like this video, don't forget to subscribe and like, and we're